Question number 48. Assertion: the length of DNA molecule is constant in a particular individual species. Reason: the length of DNA increases by the addition of nucleotides. Okay, here we have to identify the statements whichever that have been given as a true or false, and if they are true, we have to know that whether the reason statement is correctly explaining assertion statement. So let us see the assertion statement, that is length of the DNA molecule is constant in a particular individual species. So but here if you take between different species, the length will be differing, but within a particular individual species the length of the DNA is constant. So this is correct. Then the length of the DNA will depends upon the number of nucleotides present in it. So if more number of nucleotides are there, the length will increases. If less number are there, the length will be less. So that is the length of the DNA molecule is based on the number of nucleotides present in it. So the statement that have been given in assertion is also correct. Then here the two statements are correct, but this is related to the length of DNA in specific individual. This is related to the length of DNA based on the number of nucleotides, but it is not given the correct explanation for A. So question number 48, the correct answer is 2. That is A and R are true, and R is not the correct explanation of A. Question number 49. Assertion: central dogma of molecular biology was proposed by Crick. Reason: the DNA is a central molecule of hereditary storage, and Crick explains how this information is transferred to mRNA and protein. Okay, here let's come to the assertion statement here. We know that the central dogma of molecular biology. So in the central dogma of molecular biology, DNA molecule is giving rise to mRNA molecule by a process called as a transcription. And mRNA is converted into protein molecule by a process called as translation. So whatever the information that is related to the synthesis of proteins is there on mRNA, and that mRNA is getting that information from DNA molecule. So this concept was given by a scientist called as Crick. So according to the statement A, assertion statement, whatever the statement that is given is correct. Then let us come to the statement that have been given in reason. So we know that the central molecule of hereditary storage in any living organism is nothing but the DNA molecule, and the information that is present in the DNA molecule is transferred to mRNA, which will be helping in the synthesis of protein molecule. That is nothing but the central dogma of molecular biology that was proposed by Crick scientists. So both the statements A and R are correct, and R is the correct explanation of A. Question number 49, the correct answer is 1. Question number 50. Assertion: euchromatin is genetically most active. Reason: during protein synthesis, transcription usually takes place from euchromatin region. Okay, here from these two options we have to identify which of them are or both of them are correct, and whether they are giving the correct explanation of each other. So let us start with the first one. Euchromatin is a genetically most active region, and already we had discussed that in a chromatin part two regions are there, the darkly stained part and the lightly stained part. The darkly stained part is called as heterochromatin, and lightly stained part is called as euchromatin. And euchromatin is the one which is a genetically most active part in a chromatin part. So assertion statement is correct. Then why it is considered as an active part? The reason is when the transcription is taking place, when the transcription is taking place during the protein synthesis, from euchromatin region only the transcription is going on, that is the mRNA synthesis taking place. That's the reason why we consider this part to be the most active region in a DNA molecule or in a chromatin part. So both A and R are correct, and R is the correct explanation of A. So question number 50, the correct answer is 1. Question number 51. Assertion: insertion or deletion of one or two bases changes the reading frame from that point of insertion or deletion. Reason: the disease sickle cell anemia is caused due to point mutations. Okay, from that you have to identify the statements whether they are true or false. Okay, let's come to the first assertion statement. So here it is given that insertion and deletion of two bases or one or two bases, and we know that the mutations are the characters where one or two bases or more than two bases are either added or deleted, inserted or deleted from the reading frame from the point of insertion or deletion. So that is a correct statement that have been given here. So assertion is correct. And when it is coming to reason, we already discussed that in human beings, sickle cell anemia is caused due to replacement of single base pair. Like already we said that wherever GAA is there or wherever GAU is there, this base pair is replaced with some other base pairs. It may be replaced as UA, UG, UC or UU. That means when it is GAA, GAU, it should form glutamate. Glutamate amino acid have to be there in the protein chain. But when GUA, GUG, GUC or GUU, out of them any one is coming, so at that point it should be valine, that is glutamate is replacing the valine in the protein chain. So because of that one, as a change in the amino acids is taking place in the protein chain, the nature of the protein chain itself is changing. That is leading to a sickle cell anemia. So as sickle cell anemia is formed due to change in a single base pair, the change in single base pair is called as a point mutation. That means due to point mutation, sickle cell anemia is caused. So this statement is also correct, but it is nowhere related to the statement that is given in assertion, that is insertion or deletion. So both A and R are correct, and R is not the correct explanation of A. Question number 51, the correct answer is 2. Question number 52, assertion, DNA replication does not occur in interkinesis. Reason, in meiosis, nuclear division and chromosomal division occur each once. Here let us come to the assertion statement. In the assertion statement, it is given as a DNA replication does not occur in interkinesis. Actually, what is a interkinesis? Interkinesis is a gap period between meiosis 1 and meiosis 2, which is taking place during the reduction division. This is a gap period between meiosis 1 and meiosis 2. But actually, before meiosis 1, there is one more stage called as interphase. During this interphase stage, we know that G1, S and G2 phases are there. Here, during S phase, the DNA replication will be taking place, or the synthesis of DNA is taking place. So that is in the meiotic phase, during interphase the DNA replication is taking place, but not during the interkinesis. So the statement that is given in assertion is correct. Now let us come to reason statement. In the reason, in meiosis, nuclear division and chromosomal division occur each once. Here let us come to the DNA synthesis, that is the replication of DNA. How many times the replication or chromosomal division is taking place means here it is taking place only once. Whereas here the nuclear division is taking place twice, because during meiosis 1 the nucleus will divide once, during meiosis 2 the nucleus will divide second time. So that two times the nucleus is dividing. So according to that option that have been given, chromosome division is once and nuclear division is twice. So reason statement is incorrect. So question from that related discussions we can say that A is true and R is false. Question number 52, the correct answer is 3. Question number 53, assertion, in prokaryotes gene splicing is not required. Reason, split genes are absent in prokaryotes. Here from the assertion and reasons. So assertion statement talks about that splicing is not required in prokaryotic genes. What is splicing? Splicing is the removal of the non-coding region from a genetic material. So generally we know that in eukaryotic organisms the genetic material, that is DNA, contains exons and introns. Exons are called as a coding regions and introns are called as a non-coding regions. When transcription takes place, DNA is given as to heterogeneous nuclear RNA which also contains exons and introns, and then this heterogeneous nuclear RNA will undergo splicing, that is the introns are removed and continuous exons are formed in the RNA molecule. Now this RNA is called as a mRNA. So that character is present only in the eukaryotic organisms. So here the assertion statement says that in prokaryotes gene splicing is not required. Yes, it is not required because they does not have the split genes in them. Okay, so statement that is given is correct. And reason statement, split genes are absent in prokaryotes. So that is correct, and because of that reason only in prokaryotes splicing is not required. So both A and R are correct, and R is the correct explanation of A. So question number 53, the correct answer is 1. Question number 54, assertion, activated amino acids are linked to tRNA by a process known as charging of tRNA. Reason, each amino acid has its specific tRNA molecule to transfer it to the site of protein synthesis. From this we have to identify which of the statements are correct or both of them are correct. So let us come to the assertion statement. It is related to tRNA molecule. We had already discussed that tRNA contains 3 main loops, 1 tail end and 1 accessory loop. And out of the 3 main loops, 2 lateral loops are there. Out of the 2 lateral loops, one of that lateral loop will helping in identification of the enzyme which will activate the amino acid. And this activated amino acid have to be binded to the tail end, that is CCA end, towards the 3' end of the tRNA molecule. That is what we call it as a charging of the tRNA molecule. That process is called as charging of tRNA molecule. So whatever the statement that is given in the assertion is correct. That is activated amino acids are linked to tRNA by a process called as charging of tRNA. So this is correct. Then reason statement, each amino acid has its specific tRNA. Yes, how it is means, we know that on mRNA molecule the triplet codons are present, and each triplet codon is specific to the anticodon present on tRNA molecule. Anticodon present on tRNA molecule, and each tRNA contains only one anticodon. One thing that which amino acid does this tRNA bring to the site of protein synthesis means it is based on the triplet codon sequence present on mRNA molecule, and that means this anticodon, as it is complementary to triplet codon, each amino acid is has its specific tRNA, so that that tRNA will transfer that specific amino acid to the site of protein synthesis. So accordingly we can say that reason statement is also correct, but the first statement is related to the charging of tRNA, second one is related to the transfer of amino acid by tRNA to the site of protein synthesis. So both A and R are correct, but R is not the correct explanation of A. So question number 54, the correct answer is 2. Question number 55, choose the correct statement with reference to the replication of DNA. 1. Replication of DNA occurs in S phase of interphase. 2. DNA replication occurs prior to chromosomal division. 3. DNA replication occurs by semi-conservative method. 4. DNA replication occurs only once per every cell cycle. The true statements are: first one 1 and 4, second one 2 and 3, third one 1, 3 and 4, fourth one 1, 2, 3 and 4. Okay, let us come to the first statement that have been given here. It is related to the replication of DNA, and we know that replication of DNA or duplication of DNA, where the number of or amount of DNA is becoming double, that is taking place during the interphase, particularly in the S phase of interphase. So the statement that have been given is correct. Then the second one, DNA replication occurs prior to chromosomal division. Yes, the chromosomal division is taking place during the division phase. Before the division phase, the interphase will be there, and in that interphase during S of phase, the DNA replication is taking place. That means if you take the cell cycle, in the cell cycle, interphase is present and the division phase is present. During the division phase, the chromosome will divide. Chromosome will divide. Whereas before the division of chromosome only, the DNA replication is taking place during the interphase. That means for every cell cycle, first the interphase will occur, then the division phase. And during interphase, DNA replication is taking place before the chromosome division. So the statement that is given in B is also correct. Then the third one. The third statement is DNA replication occurs by semi-conservative method. Semi-conservative method means here when a parent DNA is separated, or the two coils are separated, or the two strands are separated, each of the strand will act as template to synthesize a new strand, and each of the strand will synthesize a new strand to produce two daughter DNA molecules, and these daughter DNA molecules, each of them have one parental strand and one new strand. So this method is called as a semi-conservative method, which was given by Messelson and Stahl. So this statement is also correct. Then fourth one, DNA replication occurs only once per every cycle. Yes, because whatever may be the number of nuclear divisions, either in the case of mitoid division, one division of once the nucleus is dividing. In the case of meiosis, where the nucleus is dividing twice, despite of the division of number of times the nucleus is dividing, DNA replication will occur only once in the cell cycle. So that the statement that is given in the 4 is also correct. So from the given options, we can say that 1, 2, 3 and 4 are correct. Question number 55, the correct answer is 4. Question number 56, study the following statements. 1. A DNA may consist of 50% adenine and 50% guanine. 2. A DNA molecule may consist of 30% adenine and 20% guanine. 3. A DNA molecule may consist of 60% adenine and 40% guanine. 4. A DNA molecule may consist of 40% adenine and 10% guanine. The correct statements are first one 1 and 2, second one 2 and 4, third one 3 and 4, fourth one 1, 2, 3 and 4. Here the statements that have been given are, first one, a DNA may consist of 50% adenine and 50% guanine. So let us see, is this possible in a DNA molecule? So we know that if adenine is 50%, the complementary nitrogen base is thymine, which is also equal to that one, it is 50%. But when it is guanine, so when adenine is 50%, thymine should be 50% on the other strand or the remaining part of the DNA. But what about this guanine that is 50%? So that is not possible in a DNA molecule, because when you are saying it is 50%, the cytosine should also be 50%. It is coming to 200% of nitrogen bases. So it is not possible in a DNA molecule. The total number of the nitrogen bases, whatever may be the number, it should be 100%. So this is not possible. So that one is incorrect. Next one, a DNA molecule may consist of 30% adenine and 20% guanine. We know that according to complementary base pairing, when A is 30%, thymine is also 30%, it is coming to 60% of the total amount of nitrogen bases. Then if it is guanine is 20%, cytosine is also 20%, it is coming to 40%. So the total amount or the total percentage of nitrogen bases in that DNA is 100%. So it is possible in a DNA molecule. Then when it is third one, DNA molecule may consist of 60% adenine and 40% guanine. 60% adenine and 40% guanine. So when adenine is 60%, thymine should also be 60%. So when guanine is 40%, cytosine should also be 40%. So if that is a condition, it is going beyond 100%. So it is also not possible here. Then fourth one, a DNA consists of 40% of adenine and 10% of guanine. So let us see this one. When adenine is 40%, the thymine is also 40%. It is accounting for about 80%. So A and T is accounting for 80% that is present in DNA. When G is 10%, cytosine will also be 10%. So the total percentage of G and C will be 20%. So 80 plus 20 is coming to 100%. So this is possible in a DNA molecule. So that in a DNA molecule, which of the following are correct related to that one is the second one and fourth one are possible, and these two are correct. So question number 56, the correct answer is 2. Question number 57. Which of the following types of nucleic acids do not show complementary with another molecule? Okay, here first one rRNA, second mRNA, third tRNA, fourth heterogeneous nuclear RNA that is hnRNA, and fifth DNA of 5:174 bacteriophage. The correct statements are first one 2, 4 and 5, second one 2, 3 and 4, third one 1, 2 and 5, fourth one 1, 2, 4 and 5. Solution for 57 question. The first one is a ribosomal RNA. Ribosomal RNA is single stranded. Ribosomal RNA is single stranded, and it does not form the complementary nature because it may form the pseudo helices, but it does not have that nature. So here because of the pseudo helices, wherever in the pseudo helices the complementary base pairs are coming, there is a chance of the pairing there. So there is a chance of complementary nature in this one, but not throughout the length of rRNA molecule. Then mRNA, it is single stranded, it is a straight molecule. As it is straight molecule, nowhere the coilings are there, and if there is no coiling, there is no chance for complementary nature. So it does not show the complementary nature. tRNA, it is single stranded, but as it is forming the loops, in the loops wherever the complementary base pairs are present, there is a chance of complementary nature. That is, whenever A is there, it is pairing with uracil. Whenever thymine is there, whenever cytosine is there, it is pairing with guanosine. So there is a chance of complementary nature in this one. Then heterogeneous nuclear RNA. So heterogeneous nuclear RNA is a straight molecule which is a precursor for mRNA molecule. As it is a precursor for mRNA molecule, it is also a straight molecule which is single stranded, and it does not show the complementary nature. And the fifth one is the DNA of 5:174 bacteriophage. So here it is DNA molecule, but when you say that DNA is, it should be double stranded, but it is not in the case of all the DNA molecules. So this is a special case where it contains a single stranded DNA molecule. As it is a single stranded DNA molecule, it does not show the complementary nature. So this is also not showing. Out of the 5 options that have been given, second, fourth and fifth one are not showing the complementary nature. So question number 57, the correct answer is 1. Question number 58, study the following statements. 1. That in tobacco mosaic viruses, the genetic nucleic acid is single stranded RNA. 2. In QB bacteriophage, the genetic nucleic acid is single stranded RNA. 3. In HIV, the genetic nucleic acid is in the form of 2 single stranded RNA molecules. 4. In most of the organisms, DNA act as a predominant genetic material. The correct statements are 1, 1 and 2, 2, 1 and 4, 3, 3 and 4, 4, 1, 2, 3 and 4. So let's come with the first one, that is a TMV, tobacco mosaic virus. This is a typical example for a plant virus, and in most of the plant viruses, the genetic material is in the form of RNA, that is also in the single stranded RNA molecule. So this is correct. And second one is QB bacteriophage. So in QB bacteriophage also, the genetic material is in the form of single stranded RNA. So this is also correct. Then the third one, in HIV, HIV also contains a single stranded RNA molecule, but it contains 2 single stranded RNA molecules. So whatever that is given is correct. 2 single stranded RNA molecules are present. And the fourth one is in most of the organisms, as we know that in most of the organisms the genetic material is in the form of DNA, and in very few cases the genetic material in the form of RNA molecule that we can see in some of the plant viruses. So that's the reason why. So predominant genetic material is DNA. That is also correct. So question number 58, answer is 4. Question number 59. Study the following statements. 1. RNA was the first genetic material from which DNA has been evolved. 2. According to enough evidence reports, it is suggested that essential life processes such as metabolism, translation, splicing, etc. have been evolved around RNA. 3. Some RNAs are acting as ribozymes. 4. In all organisms, the molecular structure of RNA exhibits purines and pyrimidines in the ratio of 1:1. The correct statements are: first one 4, second 2 and 3, third 1, 2 and 3, 4, 1, 2, 3 and 4. Now let us come to the statements that have been given. Let us see the first statement. RNA was the first genetic material from which DNA have been evolved. So related to this statement, we can say that yes, RNA is the first genetic material which is coming from the DNA molecule. So this statement is correct. Second one, according to enough evidence reports, we can say that all these important processes of the life, that is metabolism, translation and splicing. Metabolism includes all the chemical reactions or the metabolic processes which are taking place inside the cell. Translation, that is the mRNA which is helping in the formation of proteins by a process called as translation. And splicing, that is the removal of introns from exons or removal of introns from the heterogeneous nuclear RNA is called as splicing. All these are involved or evolved around RNA molecule. So that is also correct. Then the third one is we know that all enzymes are proteins, but there are some nucleic acids which are acting as enzymes also. Like there are some RNAs which are acting as enzymes, and those enzymes are called as ribozymes. So whatever the statement that is given is correct. And when it is coming to the fourth statement, so it is related to the RNA molecule, where it is given that purines and pyrimidines in the RNA molecule are 1:1 ratio. Actually, in most of the cases, RNA is single stranded. As it is single stranded, we can't exactly say that the number of purines and number of pyrimidines in that RNA are equal in number. When we can say that, as like in DNA, if two strands are there, one of the strand have purine, complementary it have the pyrimidine, and if it have pyrimidine, complementary it have purine. So in that case, we can say that the ratio between purines and pyrimidines is 1:1. But it is not in the case of RNA because it is a single stranded molecule. So from the given statements, we can say that statements that have been given 1, 2 and 3 are correct, and fourth one is incorrect. So question number 59, the correct answer is 3. Question number 60. A scientist has selected a double stranded molecule with 1360 angstroms length for his experiments. Due to inconvenience of its length for experiment, he had deleted 10 coils from the total DNA length by lysing the restriction endonucleases. In the remaining molecule, 20% adenine are present. Study the following data-wise statements. Options: 1. Total number of coils present in the original DNA molecule is 40. 2. Total number of nucleotides deleted from the conduction of experiment is 100. 3. Number of guanines present in the experimented molecule is 180. 4. Number of thymines present in the experimented molecule is 120. 5. Number of hydrogen bonds present in the experimented molecule is 780. The correct statements are 1, 1, 2 and 3, 2, 1, 3, 4 and 5, 3, 1, 3, 4 and 5, 4, 3, 4 and 5. Question number 60, the solution. So here in the question it is given as a DNA molecule with 1360 angstroms. Here actually we know that one coil length is 34 angstroms. As it is given 1360, it forms 40 coils. And if it is 40 coils, each coil contains 20 nucleotides. So 40 into 20 nucleotides, the total number of nucleotides will be 800. or the nitrogen bases are 800 in total. But what had happened due to inconvenience, it was cut. That means number of coils that are removed are 10 coils. If 10 coils are removed from that DNA molecule, what happens is the length will become 1020 angstroms. 1020 angstroms. Now let us come to the conclusion part. Here out of that he is saying that 20% adenine molecules are present. So if 1020 is there, how many coils means 30 coils are present. 30 coils into 20 nucleotides in each coil, it comes to 600 nitrogen bases or nucleotides, and this 600 is equals to 100%. Out of this, in the question is given as 20% of adenine is present. If adenine is 20% of this 600, the total number of adenine molecules are 120. The total number of adenine molecules are 120, and if thymines are there, so they are all complementary to adenine, so it is also of 120 in number. So total number of adenine and thymine combined, it is 240. Then guanine and cytosine. So the remaining amount that is left out, or remaining percentage that is left out is 60%. So which is 30% of guanine and 30% of cytosine. It is coming to 60%. 30% of 600 are 180, and here it is also 180. It is coming to 360. 360. The total number is 600. That is a conclusion part related to the question that has been given. Now let us come to the options that have been given here. So first one, total number of coils present in original DNA molecule. In the original DNA molecule, we said that length was 1360 and the number of coils that are formed are 40. So first option related to that one, that is 40 coils are present in the original DNA molecule, is correct. Then the second one is total number of nucleotides deleted from the conduction of the experiment is 100. So here how many coils are removed? 10 coils are removed. 10 coils are removed means when 10 coils are removed, 10 into 20 nitrogen bases or nucleotides. That means a total of 200 nucleotides or 200 nitrogen bases are removed from the DNA molecule. So in the option it is given as 100 only. So that is incorrect. So second one is incorrect related to the statement that have been given in the question. And third one is number of guanines present in the experimented molecule is 180. So already we had calculated this one. That means how many coils are left out? 30 coils are left out. In that number of guanines are 180. Already we got here. That is guanine is 30%. So 30% of guanines are there, and which is accounting for number of them is 180. So this is correct. Then the fourth one, number of thymines present in the experimented molecule is 120. So here already we said that out of 30 coils, adenine is 20%, that is 120. Thymine is 20%, that is also 120. So the number of thymines which are present in the experimented material is 120. So that is also correct. So 120 thymines, which is of 20% of the total DNA molecule, and this is also correct. Then fifth one, number of hydrogen bonds present in the experimented molecule is. Now let us conclude the fifth one, that is how many AT pairs are there? AT pairs are 120. So 120 into 2 hydrogen bonds, it is coming to 240 hydrogen bonds by 120 pairs of A and T. Then how many GC pairs are there? It is 180 pairs are present, because total number is 360. So when it is saying pairs, it is 180 pairs. So 180 into 3 hydrogen bonds between them. So it is coming to 540. So if we add the total number here, it is coming to 780. So the total number of hydrogen bonds which are present in the experimented material is 700 hydrogen bonds. So in the given option, it is given as 780 hydrogen bonds. So that is also correct. So from the given statements, only second one is incorrect. Remaining 4 are correct. That is 1, 3, 4 and 5 are correct. So question number 60, the correct answer is 3. Question number 61, the common precursor molecule involved in the production of template molecules for replication and transcription. Options: mRNA, heterogeneous nuclear RNA, rRNA and DNA molecules. So here the common precursor involved in replication and transcription, that is the template molecule they are asking about. Here template molecule is nothing but the one which is separated from the DNA molecule, and we know that in replication process, the two strands of DNA molecules are separated out, and each of them is acting as a template molecule. Where on one of the strand, that is the leading strand is formed from 5' direction to 3' direction, and the second template, on the second template, one more strand is formed which is called as a lagging strand from 5' end to 3' end. So here DNA is providing templates for replication process, where one DNA is given as to 2 dotted DNAs. Then let us come to the transcription process. In the transcription process also, we had already seen that at some place the DNA molecule uncoils, and because of the RNA polymerase enzyme, so what happens, the mRNA molecule is synthesized. So here also one of the strand of DNA is acting as a template. So here 5' end to the 5' end to 3' end, and other one is a 5' end to the 3' end. So this is the RNA molecule. In this one, this is acting as a template. During transcription, only one of the strand is acting as template, but in replication process, 2 are acting as a template. So the common precursor molecule involved in the production of template molecule for replication and transcription is DNA molecule. So question number 61, the correct answer is 4. Question number 62, which of the following is not applicable to the molecular structure of DNA? Options: A is to T is equals to 1:1. Second, G is to C is equals to 1:1. 3, A plus T by G plus C is not equals to 1:1. 4, A plus G by T plus C is not equals to 1:1. So here all the options are related to the rule prescribed by Erwin Chargaff. Chargaff had given the rule where it says that the number of purines in a DNA molecule is equals to number of pyrimidines in the same DNA molecule. So according to this one, the first one, A is to T is equals to 1:1. So whatever the number of purines or whatever the number of adenines are there, the same number of thymines are present in the DNA molecule. So this is correct, related to DNA molecule. Then second one, G is to C is equals to 1:1. So as I said that purine number is equals to pyrimidine number. Here guanine is purine, cytosine is pyrimidine. So the number of guanines is equals to number of cytosines. So 1:1 is correct. Then third one is A plus T by G plus C is not equals to 1. Actually here adenine and guanines are complementary. So if the number of adenines and guanines are equal, and the number of adenines and thymines are equal, and the number of guanines and cytosines are equal. So when it is given in such manner, so this is applicable to the Chargaff rule, that is a DNA molecule having this type of nature. When it is coming to fourth one, that is A plus G by T plus C is not equals to 1. Actually, whatever the number of adenines are there, the say equal number of thymines are there. Whatever the number of guanines are there, equal number of cytosines are there. Actually, it should be A plus G by T plus C is equals to 1. This is the correct one related to Chargaff rule. That is applicable to DNA molecule. But this is incorrect one. So, out of the 4 options, which one is not applicable to DNA structure means A plus G by T plus C is not equals to 1 is not applicable to DNA molecule. So, question number 62, the correct answer is 4. Question number 63, the ratio of purines and pyrimidines in all 3 stop codons together. Options: 2:1, second 1:2, third 3:0, 4 0:3. Here stop codons, the question is related to the stop codons. Stop codons are nothing but the termination codons or the nonsense codons. There are three types are there. Those are UAA, UAG and UGA. These are the three stop codons which are present in a code dictionary, that is a genetic code. So here let us see which of them are purines and which of them are pyrimidines. So first let us see the purines and next the pyrimidines. In this uracil is a pyrimidine, that is one is there, and both adenines are purines. Then when it is related to UAG, uracil is pyrimidine. A and G, adenine and guanine are purines. Whereas in the third one also, uracil is a pyrimidine and guanine and adenine are purines. So the number of purines are 6 and number of pyrimidines are 3. Now the ratio between purines and pyrimidines in stop codons all together is 6:3. If we simplify it, it forms into 2:1. So question number 63, the correct answer is 1. Question number 64, a scientist has isolated a segment of nucleic acid having the following base sequence: A, C, C, T, G, A, G, C, 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 G, G, G. It must be a part of options tRNA, second rRNA, third DNA, fourth mRNA. So here what is the sequence of nitrogen bases which are given here? It is A, C, C, T, G, A, and after that G, C, 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 then G, G, G. Okay, one thing here to be noted, that what is there in the sequence: adenine, cytosine, thymine and guanine. And we know that wherever the thymine is present, it is related to a DNA molecule. When it is uracil, then we have to say it is related to RNA molecule. So as thymine is present, which is present only in DNA, if you see the options, tRNA, rRNA, DNA and mRNA are given. So other than DNA, the remaining are RNAs which does not have thymine in them. So that whatever the sequence that is given is related to DNA molecule. So question number 64, the correct answer is 3. Question number 65, which of the following process represents the dominance of RNA world in eukaryotes? Options: 1. Addition of introns, 2. Gene splicing, 3. Addition of exons, 4. Arrangement of split genes. So here it is related to the dominance of RNA world in eukaryotes. So it is related to the aspect where gene splicing is done. When gene splicing is done, it is related with the dominance of the RNA world in eukaryotes. So question number 65, the correct answer is 2. Question number 66, the additional material required for packaging of chromatin at higher level. Options: 1. Non-histone proteins, 2. Histone proteins, 3. DNA and 4. RNA. Okay, we know that there is a packing of chromatin materials where the DNA is coiling around the histone proteins, that forms a bead of strings, that is nothing but the nucleosomes. If that is a normal packing that is there in the chromatin material, if additional packing is required, so there non-histone proteins are useful. So from the options we can say that non-histone proteins are the things which are required for the packaging of chromatin at the higher levels. So question number 66, the correct answer is 1. Question number 67, DNA molecule generally acts as a template for the synthesis of. Options: 1. Daughter DNA only, 2. mRNA only, 3. tRNA only, 4. Both DNA and RNA. Here it is a character related to the DNA replication and transcription process. And already we had discussed that DNA is the one which is providing template for the synthesis of DNAs. Those DNAs are called as daughter DNAs, and all types of RNAs are synthesized from DNA only. So that the DNA molecule generally it is acting as a template for the synthesis of the DNAs and also all types of RNA molecules. So question number 67, the correct answer is 4. Question number 68, euchromatin is: 1. Transcriptionally has inactive chromatin, 2. Darkly stained part of the chromatin, 3. Transcriptionally has active chromatin, 4. divide of exons in eukaryotes. So here it is related to the euchromatin part. Euchromatin is present in the chromatin, or we can say that it's a part of the chromatin where it is lightly stained part where less coilings are present. And apart from this one, this is a part which is transcriptionally active phase or active site. It is a transcriptionally the active portion, we can say, or it is a one with active genes. So from the given action we can say transcriptionally have active chromatin. So question number 69, the correct answer is 3. Question number 68, the correct answer is 3. Question number 69, in transcription unit, the promoter is located towards options 5' end of template strand, second 3' end of coding strand, 3 5' end of coding strand, 4 beside the terminator. So here when the transcription is taking place, the promoter and the terminator are present. The promoter is always towards the 5' end of the coding strand and the terminator is present towards the 3' end of the coding strand. So from the given options, 5' end of coding strand is correct related to the location of promoter in the transcription unit. So question number 69, the correct answer is 3.